Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Advancing Clinical Grade Tumor-Infiltrating Lymphocyte Therapy Manufacturing. I'm Casey Nevins, an editor at BioInsights, and joining me today is Irina Ryabinska, who will delve deeper into innovative cell manufacturing solutions for generating tumor-reactive T cells for TIL therapy within a standardized and seamlessly automated framework. Now I'd like to introduce our presenter. Dr. Irina Ryabinska is an R&D team coordinator at Miltenyi Biotec. She works on process development of personalized immunotherapies, focusing on tumor-infiltrating lymphocytes and tumor-reactive T cells. She holds a master's in medical neuroscience and a PhD in genetics. Without further ado, I'll hand you over to Irina to kick us off with today's presentation.
Thank you for the introduction, and very warm welcome to everybody joining our webinar from my side as well. Today we will be talking about clinical grade TIL manufacturing. But before we start, please briefly acknowledge my disclaimer, which provides details about Miltenyi Biotec clinical products. In today's webinar, I will give a brief introduction about what TIL therapy is, why we want to use it, and what are the challenges. Then we will talk about the clinical grade TIL manufacturing. We will show the expansion in the automated and closed system. And next, I would introduce our automated process, the full process for enrichment and expansion of tumor-reactive TIL. And we will further talk about additional TIL upgrades in the following part. We then briefly touch upon the process QC, and after that we already have the summary and our Q&A session. We know that immunotherapy has emerged as a life-changing approach for cancer treatment, and its use has improved prognosis and overall survival of many tumor types. Today we focus on adoptive T cell therapy, which has been a part of it, and we can appreciate the evolution of the ACT research by the annual publication rate increase. Talking about ACT, we can think of engineered approaches such as CAR T cells and cells with engineered TCRs on the one hand, and of patients' own naturally occurring reactive T cells on the other. The latter can mostly be found within the tumor and are therefore called tumor-infiltrating lymphocytes, or TIL for short, and this approach has recently been increasingly gaining attention. TIL therapy was initially described by Dr. Steven Rosenberg in the surgery branch of NIH. He reported the efficacy of ex vivo expanded tumor-infiltrating lymphocytes in mouse tumor models in 1986 in Science, which was directly followed by the first in-human study in metastatic melanoma in 1988. And this study has actually shown objective responses in 60% of patients, encouraging subsequent work. So let's look at the method. In TIL therapy, one needs to start with biopsy or tumor resection, and then the tumor is being chopped into fragments, and each fragment is then placed into a single well of a multi-well plate. So here. These fragments would be cultured in presence of high-dose IL-2, resulting in the TIL outgrowth out of the tumor fragments. And then TIL out of those wells, or maybe just of certain wells based on their reactivity, would be pulled together for so-called rapid expansion protocol in presence of anti-CD3 antibodies and feeder cells. And this would lead to very high expansion rates, so they basically would expand to billions, and then they need to be concentrated, washed, and reformulated, and then they are administered back into the patient. So we've talked about 1980s, but now we scroll all over to 2022, and we see here John Hahnen, whose group has actually performed the phase 3 clinical trial, and they published the results end of 2022, showing the advantage of TIL therapy over ipilimumab therapy in metastatic melanoma. And these results were so encouraging that they paved the way to the decision of Dutch National Healthcare Institute to approve the coverage of TIL therapy by the Basic Health Insurance, starting from January 2023. So here we are. Now let us look at the advantages of TIL therapy. Whereas we know that application of synthetic approaches such as CAR T can frequently lead to tumor escape, especially in solid tumors because of tumor evolution and high diversity of subclones, TIL looks very promising. And since this drug is polyclonal and different TIL clones do target different tumor epitopes. Another advantage of TIL treatment is that it is the most individualized treatment you can think of. The TILs are good at homing, and since This treatment is always autologous. There is barely any toxicity and no GvHD risks. Right, and another peculiarity of this treatment is that we do not know what the cells target, interestingly, but we do know that it works, which means that we do not need to spend time and costs on tumor sequencing, for example. So actually, this is a very straightforward method. However, when we look at this in historical time perspective, we see that whereas CAR therapy, which emerged later, has already been translated into several commercially available drugs, TIL therapy has not been commercialized state today after almost 4 decades. And this is partially because of high costs and complicated manufacturing procedure. Well, I need to note that we do hope for the first TIL-based drug approval early next year. Talking about complicated manufacturing, let us look at this process step by step once more. It starts with multiple plates, then cells are moved into T flasks, then to a bioreactor. They need to be fed under the hood and cultured in the incubator and later washed in a cell washer, meaning this includes multiple devices. And each of those devices brings certain variability, let alone the operator-related variability, so it is generally not robust. Another challenge to TIL manufacturing is high contamination risk, as this process includes multiple open steps. And I brought here an example from a publication reporting TIL generation out of a cohort of patients where 28% of cases, in 28% of cases, they actually had to discard the culture due to contamination. As you see here on the left, and in 14% of cases, this is the next bar, TIL just wouldn't grow from the fragments. So there's definitely a risk here. And finally, the last bar, in the remaining 57%, there was outgrowth. But for 25%, here in the middle, there were 3 or less wells out of 12 outgrowing, meaning that this outgrowth might be not sufficient, so it would compromise the expansion. The TIL manufacturing is definitely very laborious, and it relies on highly trained staff. And finally, this process is also very time-consuming, because if we are thinking of TIL outgrowth alone, taking on average 3 to 5 weeks, followed by the 2 weeks of rapid expansion, this is the time not every cancer patient can afford. A couple of years ago, here at Miltenyi, we came up with the idea to combine this very promising but also complex and sophisticated therapy with Miltenyi's great expertise in cellular drug manufacturing. And I just note here on the side that our technologies have been used in more than 180 clinical trials in 45 countries and in more than 360 cell and gene therapy companies worldwide. We already had run-in solutions for graft engineering, manufacturing of NK and CAR-NK cells, as well as monocyte-derived dendritic cells, and finally many others as well. And Miltenyi, of course, has a great expertise in automated and GMP-grade manufacturing of engineered T cells, such as, for example, CAR T cells. And therefore, adding TIL to this portfolio was very exciting. In the upcoming slides, I would like to explain how we developed an automated TIL manufacturing process, as well as show you some data. But first, let me introduce to you our platform, in case some of you might be not familiar with it. So here in the middle, you see the CliniMACS Prodigy, which is a powerful instrument that can do any kinds of cellular processing operations. And this includes seeding, cell activation, washing, expansion, concentration, centrifugation, transduction, magnetic selection, and so on. So literally anything you would like to have. And this way, it enables end-to-end cell manufacturing, fully automated, enabling increased reproducibility and standardization. And the complete cell processing for each patient is done in a single-use tubing set, so it is closed and sterile. And as a part of a package, Miltenyi provides GMP-grade reagents to support our processes. Furthermore, CliniMACS Prodigy is equipped with a very user-friendly software, which ensures automated documentation of the manufacturing procedures. We started our development with automating the rapid expansion part of the protocol. And for this, we had to ensure that we have all the components of the system, including the GMP-grade medium, activation reagent cytokines, the tubing set, and of course the software app specifically tailored to TIL manufacturing needs. The cartoon on the left shows automated feeding of the cells, but what I actually want to show you is our expansion performance. So here you can see that we were able to expand young TIL from melanoma patients from around 1 million or even less to several billions, and there was a high fold change in our final products, and they had very good viability. We also compared our automated expansion to manual culture and have observed significantly higher expansion rates and comparable viability of the TIL. Finally, we were interested in characterizing the products. Obviously, working with the TIL, one would always have the donor-to-donor variability, but we were quite pleased to see only a small fraction of our cells to be positive for multiple exhaustion markers, such as PD-1, TIM3, and LAG3, and the majority of the expanded TIL had central or effector memory phenotype. In the course of our development, we came up with several recommended culture regimens, which an operator could select if unsure about their feeding and medium exchange schedules. And selecting one of these protocols, one would need to consider starting cell numbers, as well as whether transduction step is desired or not. However, as you know, every patient is different, and there might be cases where one would like to modify the cultivation schedule performed by the machine, and this is of course possible before the culture as well as any time during the actual process. One can always add, remove, or modify activities, which could be not only medium addition or exchange, but it could be also, for example, activation or deactivation of cell shaking, which can also be done at different intensities. And this can be useful once the cells reach higher cell numbers. Despite the actual work in this case is performed by the machine, we of course want to have a full control, and for this our process has a sampling button which can be pressed anytime during the culture and will result in a sterile sampling from the chamber where the cells are cultured to the small sampling pouch that you can see here on the right, and this can be then welded off and taken to the QC department. And here you see the example of our glucose, lactate, and viability measurements during one of the runs, as well as some information about the culturing volume, which can be changed during the culture, and the shaking intensities that we programmed. The CliniMACS Prodigy is also equipped with the microscope, so I'm always tempted to take pictures of my expanding TIL. And the nice part is that the device is automatically saving all the pictures I take during the process and add them to the process protocol folder. Whereas we performed most of our development work with melanoma-derived TIL, since it currently remains the leading indication for the TIL therapy, we also performed our automated process with TIL from other indications, such as pancreatic, renal, colorectal, and breast cancer, and we're quite happy to observe good expansion and viability. We are of course encouraged by our first results. However, at the end of the expansion, we usually achieved up to around 4 billion cells. And as we looked at the ongoing clinical trials, most of them were aiming to have higher cell numbers, 1 billion being rather the lowest border. And I have brought here an example from the phase 2 clinical study performed by the current TIL market leader, Iovance, which was published last year, where they report to have injected on average 21 billion of cells. And I think what I didn't mention in this webinar is that the standard Prodigy culturing chamber has a volume of 400 milliliters, of which we only can use up to 250 milliliters. So there was most probably a space limitation. And in order to overcome this hurdle, we had to switch our process, which now would be the next version of the process and is in development right now. So we had to switch the process from the tubing set we were using, the 520, to another tubing set, 620, which was bearing a larger chamber. And with this, we were able to achieve higher cell numbers in the range of 1e10. To ensure that the chamber size has not affected other parameters, we of course assessed the viability and the phenotype of the expanded cells, and we're happy to find similar results to the ones we have observed in our initial development. We were also looking into the possibilities of achieving even higher cell numbers in the range of hundreds of billions, and in this case, this can be done by the connection of the Prodigy to external culture vessels with even larger volume, whereas the cultivation automation will still be taken care of by the Prodigy. But interestingly, discussing our development with our customers and partners, we realized that there is a high need for harvest automation for the TIL product, specifically when it is being cultured to these huge numbers and correspondingly high volumes. And the good news is that we have a solution to that as well, also Prodigy based. And this is the app called CliniMACS Prodigy Concentrate, which allows to concentrate up to 50 liters of cultured material down to as few as 60 milliliters or more. This is actually user-defined. And the application is quite flexible. It also permits to adjust the amount of washing cycles, the centrifugation speed, and the flow rate. And it turned out to be very handy because we found out that we can concentrate, for example, hex cell cultures at any flow rate and get the same recovery PBMCs, for example, and this would potentially apply to TIL as well, are more fragile and show good recoveries upon concentration at lower flow rates, but it decreases. The recovery would decrease if we flush them through the pump at a rate of, let's say, 150 milliliters per minute. We mentioned earlier that the conventional TIL expansion procedure bears some challenges, such as, for example, the time factor, and I would like to take a step back and look into this again. The time needed to expand TIL is dictated by the enduring and delicate fragment outgrowth procedure, as well as the need to reach extremely high cell numbers, explained by the fact that only a fraction of those cells are truly tumor-reactive. Most of the cells are just bystanders and might be, for example, coming from the bleeding during the surgical tumor removal, or just be not tumor-specific, meaning that there is also a risk of low therapy efficiency associated with the expansion of inappropriate TIL clones. Solution to this problem might be an early pre-selection of the tumor-reactive T cells. Whereas back then in 20th century, the standard TIL protocol would include a step in which outgrown TIL would be again co-cultured with autologous tumor material to select the wells in which interferon gamma production was registered. Now, in 2013, the group of Daniel Powell in the University of Pennsylvania suggested to select the TIL based on the CD137 expression after a short incubation of the tumor digest. CD137 is known as a T cell activation marker and was reported to accurately identify tumor-reactive T cells. So we thought to take advantage of this finding and include the CD137 TIL enrichment directly from the tumor digest into our automated process. This step would allow us to automate TIL manufacturing from the day of the tumor resection and digest to the day of harvest and infusion, and would substantially shorten the total manufacturing time. Besides saving the vein-to-vein time and ensuring completely closed manufacturing process, we would now also expand only tumor-reactive and thus the most potent TIL, and can afford lower cell numbers, which would be less stressful to the patient and more cost-efficient. Of course, to allow for such pre-enrichment step in the clinical grade, we again had to make sure that not only the culturing but also the enrichment reagents are provided in the corresponding quality. We were also using the in-house tumor dissociation kit and the GentleMACS tumor dissociator. Having assembled the reagents, we were lucky to obtain melanoma tumor samples. So we had digest and performed several experiments with the full process, including digest stimulation within the Prodigy chamber, labeling and enrichment of CD137 positive cells, as well as subsequent expansion. And here you can see our enrichment purity and representative flow data. In some cases, the samples were quite small, so we could only get a few thousands of TILs after the enrichment to start the expansion with. And despite that, in most of the experiments, we would still get billions of cells after the 2 weeks of the rep, with quite remarkable fold change. Also, after the full process performed with TILs enriched from the tumor digest for CD137, we obtained highly viable products with high fraction of CD8 positive cells, which is also associated with good therapy responses in the TIL field. And furthermore, we again saw most of our cells to have the memory phenotype and rather low expression of exhaustion markers. As we discussed in the beginning of this webinar, polyclonality is the key feature of the TIL drug, and there is always a concern in the field about whether we are favoring certain clones by the expansion while losing the others. To address this, we have expanded non-selected bulk TIL on the one device, whereas CD137 enriched product was expanded on the other device in parallel, and finally the negative fraction or CD137 depleted TIL was expanded on the third device. We then analyzed the TCR sequencing results from the three products as well as the pre-expanded TIL. And we observed high clonality in all the expanded fractions compared to the TIL before expansion, which is generally known and observed phenomenon. But interestingly, though, the CD137 enriched TIL product contained more clones with lower indices compared to the bulk and negative fraction. When we looked at the morocity similarity index between the products, we found that the CD137 positively enriched TIL had the most distinct TCR repertoire out of the three products, whereas the unselected TIL and the negative fraction were much more alike. So finally, we compared the TCR repertoires for the expanded products to the pre-expanded TIL, so the TIL before expansion, and found the highest overlap index values of the CD137 positive fraction together with the pre-expanded, suggesting that the other fractions were losing the clones and unspecifically expanding bystanders. Of note, recent data from the group of George Kokos in the University of Lausanne reported high preservation of TIL clones originally found in the tumor to be associated with TIL therapy response. Next, we wanted to assess the functionality of our TIL product and also confirm the advantage of the CD137 enrichment step. So we co-cultured the CD137 enriched as well as CD137 negative fraction with autologous and allogeneic tumor cell lines and measured interferon gamma production. On the left side, you can see the representative flow cytometry plots with a profound response among the enriched fraction, whereas looking at the negative fraction, we see almost no reactivity. And on the right side, there is quantification of these results, where we can appreciate that the response was actually specific to autologous cell line and was not observed upon co-culture with allogeneic cell line. In these co-culture experiments, we also measured the levels of CD137 itself. And I need to note that during the rep, in the presence of CD3 signal and feeder cells, CD137 goes down over the expansion time, and therefore the baseline level of CD137 expression was actually similar between these two fractions, the CD137 enriched and the negative. But as we co-cultured them with the tumor cells, we could once again see the same effect, or even stronger effect than with interferon gamma. So what you can see here is the strong reaction within the CD137 enriched fraction, whereas the negative fraction showed the expression levels similar to the background. And also here we can observe specific response to the autologous tumor cell line, as you can see on the right side. We discussed that successful application of TIL therapy in patients was demonstrated in 1988. So since then, there was, of course, a lot going on in the TIL field. And one of the research directions was aiming to find features of the most efficient TIL product and to validate these findings by sorting the TIL based on certain marker combination. Also, many strategies were focused on pre-selecting the T cells first, or for example, only CD8 positive cells, which I believed to be actually more efficient TIL population in this case, and further sorting this population based on various marker combination. And the marker preference was quite dynamic, with several combinations reported to be the most precise. So we thought of ways to enable the flexible sorting within our clinical grade process on the one hand, and we still wanted to maintain the process sterility and quality on the other. So to make this possible, we decided to include the MaxQuant titer sorter into our pipeline, taking advantage of the closed sorting system and availability of GMP-grade single-use sorting cartridges. As a proof of concept, we sorted for CD4, CD8, and CD137 positive TIL. And you can see our sorting strategies here. So we first would be sorting for CD4 and CD8. Well, we first would be gating for CD4 and CD8, and you can see this complex 8 kind of shape gating. And then the resulting cells would be additionally gated for CD137 positivity. And you can already see from the representative plots from the input, the non-sort, and the finally sorted fraction that the performance of the sorting was quite high, and we could basically deplete the cells we didn't want to have and enrich the cells within our gating strategy. And also we could be happy about the high yield of over 85%. So to continue with this, we expanded the sorted TIL subset as well as, of course, the non-sorted control in the CliniMACS Prodigy with the rapid expansion protocol. In both cases, we started with around 1 million, actually a little bit less in the tighter sorted fraction, and then after 2 weeks, we harvested around billions of cells. The numbers were more or less similar between the control and the sorted TIL group. After the expansion, we harvested the products and wanted to validate the functionality of the tighter sorted TIL. So we performed the co-culture assays with autologous tumor cells and compared the tighter sorted cells to the bulk TIL. We can see that higher percentage of the sorted cells depicted here in the dark blue produced interferon gamma after the co-culture with autologous tumor, and similarly higher fraction of sorted TIL upregulated CD137 and also accumulated CD107, as you can see here on the right, compared to the non-sorted control. So that suggests maybe a higher reactivity on the one hand, and also, as we can see from the same experiments, the incubation with allogeneic tumor didn't yield this reactivity, so suggesting the specificity of the response. Another trend in the current TIL research is aiming to improve the TIL product through genetic engineering. Here we can identify several directions. For example, we know that TIL growth in vitro as well as in vivo is very cytokine dependent. We use high concentrations of IL-2, or people also use alternative cytokine combinations in order to expand the TIL, and the patients receiving TIL also need to be injected with high doses of IL-2 at very intense schedule, for example, every 12 hours in the first days. In fact, while TIL therapy is considered to be safe, most of the adverse events occurring in the clinical trials with the TILs are attributed to the accompanying IL-2 treatment. Thus, cytokine-independent survival would be one of the top desired features of the next generation TIL. Another important direction is combating TIL exhaustion, as coming from a tumor where they have been continuously stimulated for a prolonged time period, TIL are certainly much more exhausted compared to, for example, blood-borne primary T cells. And this is directly from the isolation. Therefore, researchers are working on generating TIL, for example, with knockout for certain exhaustion markers and checkpoint inhibitors. Also, similarly to CAR T cells, TIL also face the hostile tumor microenvironment and need to resist that. So this becomes specifically critical when trying to apply TIL therapy in a wider range of tumor indications. One example of the work to improve TIL resistance is, for example, generation of TIL deficient for TGF-beta receptor 2. Finally, there is also work towards generating an even more potent TIL, introducing the CAR structures, the CAR constructs. This work is being done using various technologies such as TALEN, CRISPR, or viral vector delivery, just to name a few. And luckily, the CliniMACS platform enables both viral and non-viral delivery of genetic material. So on the one hand, you can see here on the left, the Prodigy culture, and specifically the expansion culture within the TRT process that we are using for the TIL manufacturing, has these optional activities which one can always program. And one of them is called transduction, which ensures automated and close delivery of the virus material with, of course, any desired enhancers into the chamber with the cells. And another activity is called spin, and this enables spinacylation with the operator-defined parameters. For example, there is flexible spin temperature. And we are able to successfully transduce TIL within the Prodigy, and we have currently more work in the development. Now, on the other hand, the CliniMACS Prodigy can be actually connected to the CliniMACS electroporator with software and tubing set shared between the devices. While TIL electroporation is a part of our current and future development, so I couldn't share data, but we have good experience in other TIL cell electroporation, And there are also partner and customer publications reporting car production using our Prodigy and electroporator combination. Coming to the end of my talk, I would like to briefly get back to the challenges of conventional TIL manufacturing that we defined in the beginning of today's webinar. We talked about process variability, contamination risks, complexity and heavy dependence on the trained and experienced personnel, and we were also talking about the process time. By introducing the automated TRT process on the CliniMACS Prodigy, we addressed these challenges by replacing the multiple devices with a single CliniMACS Prodigy platform, and we were also able to abrogate the contamination risks, which are basically zero in the Prodigy platform, whereas there is a variable amount reported by different publications. We were also able to free hours and maybe days and weeks of sterile bench work. And this small bar here on the right, a little bit on the above, is actually demonstrates the different process steps in which operator needs to take attention on the device. Those are shown in the pink, but actually this is just day -1 and day 0. So this is the process setup. And also at the end of the process, all the other days, it's pretty short, just taking a look at the device and one is free. And the blue part of this bar actually is showing the steps of the process the device is managing on itself. Right, in fact, according to our calculations, which you can see here on the lower graph, the operator, which still needs to set up and control the automated manufacturing, spends around 5% of the total process time in the preparation work and also in front of the device. And this number gets slightly higher, just slightly higher, if we include the time for the flow analysis for the IPC and QC samples. And finally, considering the full process time, if we directly extract and enrich the TIL from the tumor digest, our automated TIL manufacturing protocol is actually the shortest of currently disclosed TIL manufacturing protocols in the field. Now, let me spend just another slide to show you a very brief overview of the in-process and quality control steps required for a smooth TRT process workflow. First, we need to determine the characteristics of our starting material, which might be very variable. So we need also the cell numbers that we are starting with. And those need to be entered as an input parameter and will be protocoled by the device. Before and after CD137 enrichment, the levels of CD137 are assessed by the flow cytometry to look at the purity and recovery of the labeled cells. And later, if there is a genetic modification step planned in this process, there is, of course, a control step for the transduction efficiency. And there is a series of subsequent controls during the expansion just to assess the expansion rates and also to potentially modify the culture regimen if required. And finally, in the end, there is a big final QC procedure that needs to be performed to check if the product meets all the release criteria. To assure full support of our automated TIL manufacturing process, including the intra-process control steps and the quality control, we have developed special protocols and antibody panels for the flow cytometry analysis. Within the MAXQuant software, we also have express modes, which allow analysis of certain flow panel data, such as, for example, immune cell composition, in an automated fashion. With this, I would like to summarize my presentation. Today we talked about TIL therapy as highly promising adoptive T cell therapy for solid tumors. We also took a look at an automated way of such therapy manufacturing and ensured that the automated manufacturing yields robust and reproducible results and also helps to overcome multiple hurdles of conventional manufacturing protocol, generating improved quality drug at shorter time. We also discussed trends for TIL of the next generation and have seen how special steps such as multiparametric sorting and genetic engineering can be included into the automated TIL manufacturing process. Finally, we mentioned availability of supporting protocols and documentation, including the in-process control flow panels here at Miltenyi Biotec. And what I forgot to mention is that we also provide certified trainings for the TRT process. This brings me to the end of my talk. Thank you for your attention, and I'm happy to take questions.
Thank you so much, Irina, for a great presentation. So now we're going to start our Q&A. Our first question is, how is the tumor processed and pre-REP culture done before the initiation of the REP process? Also, is the enrichment process done in a closed system?
Thanks a lot for this question. Right, so the process allows both ways of doing that. We've discussed in the webinar that you could do only the expansion on the Prodigy, and you could also do the complete processing, meaning that you start from the digest, you do the enrichment, and then you can continue with the rep. In the case of the last variant, there is like no pre-rep. There is literally a stimulation culture, which is usually overnight, but you can do something else that depends on your wish. The process is flexible and it allows the operator to program different culture times. On the other hand, if you say, I don't want the enrichment, I really want to outgrow the TILs from fragments, then in this case, the fragment culture is actually done outside of the device. We have in our to-dos to automate also the fragment culture. Right now, it's not, let's say, not been verified. We have certain concerns, but we are pretty sure we can overcome them soon, or maybe just test and see that it happens. That's why, yeah, there are basically two options. You either do it like our way, let's say the suggested way, then you don't need the pre-rep. And if you do it with the fragment outgrowth, then it's small scale. A small comment, the enrichment is done in a closed system, obviously. That's part of the Prodigy device, and the part of the tubing set is the column which is put onto the magnet in the Prodigy.
Great, thank you so much. Our next question is the automated Prodigy TIL expansion process feeder cell free?
Oh, thanks. That's a good question. Well, the data that we've shown is not feeder free, so we are using the feeders. Those were irradiated feeders coming from leukapheresis products. We usually mix several donors. We do freeze the LPs and then we thaw and irradiate. You can also do it the other way. We have tried that. That works as well. You can irradiate the mixed LPs, freeze them, thaw, and put into the machine. Both works. We have customers who consider or who claim they would love to do it feeder-free, and I think who are doing it feeder-free. I cannot say more because this is the customer work, but there's definitely nothing where the process would limit you and say, now you need to add a feeders, or now you're not allowed to add them. So there's an activity which you can program. You can schedule it and say, okay, at 3 p.m., I don't know, on the day 0, on the day 1, I'm adding the feeders. And that's where the device will show you the hint and say, now, please weld on the back with the feeders, enter the starting material, like all the information about the feeders, and then they will be added to the culture.
Great. Our next question is, how many days were used for the pre-REP process before the initiation of the REP process in your study?
Right, so for the pre-rep, again, if you're doing the fragment outgrowth, it's up to you how long you do it. In our examples, that really differed from samples to samples. For melanoma, it's shorter, it's a couple of weeks. We've done some studies with other tumor types. As you've seen, there's like pancreatic, breast, renal, and so on. For pancreatic, it took a while. We had some which were 3 weeks old. We had some which were older. They all did grow in the end, but that lies on the side of of people doing the small scale.
Thank you so much. Oh, sorry.
Yeah, if you do the full process, if you do the full process, like with the enrichment, we can like come over it. So we don't do any pre-rep.
Great. Sorry for cutting you off there. Our next question is, when will the larger chamber be available commercially?
Thanks for this question. Well, that's a great question. The large chamber is available right now, and there's nothing preventing you from working with that. The only reason why we say we are actually in development is because currently the large chamber is used in the released process in the TCT-LS. That's the process which is used for car manufacturing. That includes CD4 and 8 enrichment, followed by the expansion. Nobody prohibits to be using it for TIL. But what we are planning to do is we are planning to issue a separate process, which will be the software for the TIL, which would include the CD137 enrichment on the large chamber and then the expansion. But right now we actually do have customers using the last chamber for and the TCT process, which is available there for culturing of TILs.
Great. So we have another question about the feeder cell line. This question says, are you using specific feeder cell line?
That's a great question. Thanks for that. We've not been doing that because there are, well, to be very honest, because there are patents all around that, and we can definitely buy the cell line and use it, but to show the customers that the system is working, we don't need an extra cell line. We show that it works with the basic settings, like the very, very classical ones. We also use IL-2, if you've noticed, which is the classical protocol, but not the only one possible. We have tried other things, and we know that they also will work on the device and with the software that we provide.
Our next question is, I would like to ask if it is possible to start the process with frozen material.
Thanks for that question. Well, that's definitely possible. We have tried that. We have tried frozen outgrown TILs. We have tried frozen tumor digest. We have even tried to digest from frozen pieces. That works as well. Yeah, in terms of tumor pieces, I would suggest to first digest and then freeze, because you might not like the work with the frozen tumor pieces, but otherwise you definitely can thaw the frozen material and then put it on the Prodigy and start the process.
Great. Another question about the feeder cells. What type of feeder cell is preferred for the Prodigy TIL process? Irradiated PBMC or an engineered cell line?
I see feeder cells is a hot topic today. Well, I can definitely understand that. We have not done a side-by-side comparison between irradiated PBMCs and the engineered cell line. We do know from publications that both works. Here in-house, we were doing irradiated PBMC, actually irradiated LPs. We just freeze and thaw them, so we actually don't do the lysis in this case. Right, so I cannot really address that question, but I'm pretty sure that both would work more or less equally.
Great. Next, we have, after REC, do you perform any reactivity tests? IFNY, for instance.
Well, but that's for sure. Thanks a lot. I mean, the question is nice, and I think we can, if the slides are shared later, or I don't know, I can also otherwise explain with words that we did perform that. We also did show it here, but we definitely have more data where we do incubate the TILs with autologous tumor cell line. For that, you obviously need the tumor cell line to be available. And then you can co-incubate, and we measure interferon gamma release. That's what is done. We've also measured the other cytokine release. That does work.
The next question is, in our lab, we are not using CD137 enrichment, but we considered selecting the T cells out of the digest and potentially enriching them later in the culture period for our markers. Is this possible?
Right. You can see multiple ways of doing that. So first of all, I'd like to point out that we have this titer connection, so you could potentially connect the titer sorter. If you have one. Well, in any case, there is a possibility, and it's also a sterile one. So there's a closed sorting and the closed Prodigy. You can connect those two, and we have available variable markers available for the sorting. And the advantage of flow sorting, or of titer sorting in our case, would be the possibility to connect multiple markers, and do any kind of gating. On the other hand, if you would like to do something else than CD137 on the Prodigy, you might have that option as well. Well, for example, we do have a process which does 4 and 8 selection, you know that. The TRT, as we developed it, does CD137 enrichment, and if you consider different markers and you want to use the column, the magnetic, and you don't want to use the flow sorting for some reason, then one can potentially design a cap, like a customized application, which is then when you need to contact us, and then we come towards you with our solutions.
Thank you. It seems CD137 is also a hot topic. The next question says, does the CD137 enrichment include all tumor-reactive clonotypes?
Well, thanks a lot for that question. That's a good one. I can tell you that it's hard to address that question in terms of maybe you could specify what kind of test you would prefer, but the functional studies that we have shown in this webinar, where we compare the negative fraction and the positive. So the one which was selected was also the CD137. We co-incubated with autologous tumor cells, and then we take the ones which were kind of depleted. So everything which went to the second bag during the magnetic sorting. After the co-incubation, we see the signal. So we see interferon gamma release, we see the upregulation of CD137. Out of the positive fraction, we don't see more or less anything in the negative fraction. Therefore, this would suggest that you kept all the reactive clones in the CD137 enriched fraction.
Great, that's a great answer. Next, we have, do you have a solution for sorting markers inside a cell?
Do you have a solution for sorting markers inside the cells, the ones which are not on the surface?
I don't know. That is all they said.
Well, I think as long as you need to fix the cell and label the markers, you will be losing the cell. It will not be viable anymore. So that is technically challenging. Maybe if it is possible to specify the markers, we could think of it together.
Yes, they can always email you after the webinar is over, and then
Oh,
you
for
can
sure.
Email. Yes.
Yeah.
So our next question says, you mentioned that you stimulate cells overnight, but in my experience, we've done it longer. Can I also do it with your process?
Oh, for sure. That's a good question. I know of people doing different things. The process can be programmed in a way that when you do the pre-enrichment culture, you have also that flexible matrix, which we have shown for the expansion culture. It more or less looks the same. So you can add, remove activities. You can also schedule the end of culture for any day, like from 2 to, I think, 19 or 21. So there is a restricted amount of days, but it's definitely far beyond 1 or 2 nights. And that's how the operator can decide what they can do.
Great. Next, we have, do you have data on the rate of unrecoverable instrument failure during processing? What is the main cause of failure?
The instrument failure. Well, thanks for that question. I think it refers to the technical performance of the machine, like if machine is, well, important is to mention that our machine relies on the constant power supply. So if there is a risk of let's say, power outages in your facility, then we strongly suggest buying basically a battery or somehow, so like an external power source, which you could connect to the Prodigy. I do know of customers using that, and that works smoothly for them. The main thing is to remember, to recharge the battery. That happened as well. But once that is fixed, the Prodigy is actually doing quite well. What one can think of, if you have the cells that are strongly clogging, we do have filters in our bags. So when you put the starting material into the bag, there are 2 filters. Then after the labeling, the cells go through the filtration bag, and that one also has filters specifically for the cells that are clogging, because we don't want the column to get clogged. We don't want the access to the chamber, which is a thin cannula, to get clogged as well. So we need to keep an eye on that. If it gets clogged, there are actually troubleshooting scenarios which can be applied, and then the process can be rescued, but that will be associated with time delays, for example, where you can see your sensitive cells losing viability and so on. So that is something to get control of. What other issues? Yeah, well, take care of your device.
That is definitely all very helpful advice. Thank you. Next, we have, can another marker besides CD137 be used for TIL enrichment in the Prodigy?
Well, good question. I think we have potentially touched on that topic in one of the previous questions. That might be. So we have, I think it's important to say that we have released processes. the fact that they are released means that they have certain status, they have been verified, the software has been verified, the process has been validated, we have the performance data which we can show, and the processes are ready for, let's say, GMP-like applications, meaning that you have quite an easy time applying with usage of this process to various kinds of authorities. I can speak for Europe, that's definitely a very good case to go. I think in the US one would need a couple of additional documentation or like in-house tests, but that's still quite nicely prepared on the Miltenyi side. When you want to do something else, you can go for a customized application where you ask us to do that work for you with a different marker, or you can do something in-house where you just, basically you can replace a vial with a different vial, then it will be non-validated, which will let you have the issues with authorities potentially, but technically you can also do it for your research purposes.
Great, thank you. Next we have you tested to genetically engineer TILs? And if so, how?
Okay. Well, thanks. That's definitely a very interesting area, which I think, many people would like to know more about. Genetically engineering TILs, as mentioned, that would include either viral transduction or something else. We can do electroporation. We've not done in-house electroporation of TILs yet. We hope to do so. For now, we have transduced TILs, and that has yielded quite good results, so we are happy with that. Well, one can look at using transduction enhancers if one want. One can also try without. One can play with different time points. But we have tried many things that actually works nicely.
Great. Our next question is, can I use Transact instead of feeder cells?
Okay, thanks for that question. I've been hearing that a couple of times already, that people would prefer Transact to feeder cells. We would maybe not recommend it immediately, but I know of people using that, and it seems to work for our customers. So that's something one need to explore. I don't have much experience here in the in-house, in the R&D side, but that's something which I think people do.
Nice. Okay, so our next question refers specifically to one of your slides. It says, in one of your slides, it is written that you can use the tumor or leukapheresis as a starting material.
Right. So we do have an option. Let's say when you start the TRT process, you can actually select between either tumor digest, the starting material, or leukapheresis. For leukapheresis, it's understood that if, let's say, you want to capture some reactive cells, which might be even virus reactive cells, so whatever reactivity you would prefer to see there, in that case, you would need to add certain stimulus to the leukapheresis product. The leukapheresis scenario of the process is slightly different, so it has slightly different washing schedule, because basically loading leukapheresis to the device would require additional washing. And then there is a mandatory stimulation step where you are prompted to add something to stimulate the cells, and then they will be selected for CD137, and you can also expand them. That works pretty nicely. We tested that with virus a lot. We have good results. You can think of something else, so it's up to your creativity.
That's great. Thank you. Our next question says, how long do you lyse the tumors?
Okay, great question. Thanks a lot. We do lyse tumors with CliniMACS because that's the in-house device. That's what is available to us with also, with this tumor dissociation kit. We do it for 1 hour because there is a protocol available in the CliniMACS. I think it's one of the standards, which is for the tumor prep, for the tumor lysis. It's 1 hour long, so that's what we are using.
Our next question you may have touched on already because it's about feeder cells, but it says, which feeder cells do you use and at which ratio are you adding them?
Okay, that's an important question, by the way, because there are multiple ratios published, and I think also various parties would argue for various ratios, low and high. What is important for the Prodigy, because we know of different functions of the feeder cells, and one of them is to provide certain cell-cell contact to to the TILs. Now, as we do select TILs, actually, even if we don't select them, we usually start with very low amount of material. So the cells would like to feel, to feel the cell-cell contact, and for that, in the Prodigy chamber, which is pretty large, so it's like 100 square centimeters, we use the fixed amount of feeder cells, so it's always 48, like 400 million. And that's what works. So we do that independently of the amount of TILs that we seed in the expansion culture. Therefore, the ratio would differ from starting material to starting material. We recommend starting with 1 million for the expansion culture. That's what nicely worked in the verification. We do know that more and less does work as well, but that was the average that we found to be nicely working. And in that case, the ratio is 1 to 400.
That is great information. Thank you so much. Our next question says, can I monitor the process via the internet?
Oh, great question. Well, actually, you cannot. So the Prodigy can, it has a board computer, so to say. You can connect via a USB device to retrieve all your protocols, to retrieve your microscope pictures. That all is possible. What you cannot do is you cannot connect via internet. It had an advantage. So a couple of years ago, I think there was like a hacker story where everybody was attacking certain parts of the code and different medical devices, and the Prodigy was safe because not being connected to internet. I mean, that's a true story, by the way. But obviously, people would like this functionality. What is, however, possible is to attach an external alarm device to the Prodigy. So that is possible, meaning that anytime the Prodigy would need attention, because sometimes it just turns on and say, well, I don't know, exchange your waste bag or something. If you program that in the activity matrix, if you didn't, it will not. But it's suggested to program that. Or also, let's say if you forgot to exchange the waste bag and you programmed multiple feeding activities, and at some point your waste bag, which is 5 liter large, gets out of volume, then you get a push alert and you say, waste bag is full. So if the Prodigy is doing that and you connected the external alarm, it will, I think, send you an SMS or like give you a signal and tell you that you need to go to the clean room. So that is possible, but complete monitoring of the process or like guiding the process through internet is not available yet.
Got it. Thank you. Our next question says, do you know in current clinical trials, what is the typical cell dose?
Well, that's very variable. As a rule, people start with something like 1 billion and go up to 111 sometimes. And average, it would be several billion. I would say it actually goes rather into the direction of 1E10. That's why we initiated the work with the large chamber. But what I hear from the conferences and from people reporting about their trials is that they try to outgrow what is possible. And if it was a few, they inject a few. And if it was more, they don't throw a half away. So they do use everything they cultured.
Great. Well, thank you so much, Irina, for answering those questions. Unfortunately, that is all we have time for today. So any questions we didn't get to, we will reply to by email. All that's left is to thank Irina once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.